Next, I would like to introduce Kalani Carlson. We're going to talk about understanding variable macroalgal dissolved organic carbon dynamics with diasporic ʻŌiwi research methodology. Take it away.
Mai ka ko o wau o Kalani e kana ko iwiao na aina e mai apa o hilawai ke mai ku tuta kane ma I kia manawa ke noko ni au maka aina o ke kanaka ainu e no I ana au I ka limu ma ke kula nui o Hokkaido. Aloha everyone, I'm Kalani. I'm a PhD student researching seaweed biogeochemistry. I was born and raised in the international diaspora, but my puna Hawaii are mainly from Hilo. So really happy to be here for the symposium and to spend time with my ohana. I'm doing my research through the Japanese Settler Colonial Institution of Hokkaido University. And I'm focusing on this kelp, Hosome kombu or Saccharina japonica var. Religiosa. So this is in Oshodo Bay in Ainu Moser, or the indigenous homelands of the Ainu people. And so my topic today is going to be on understanding the variability in the dissolved organic carbon dynamics from macroalgae, specifically looking at this kelp, but also thinking about some corollaries to other macroalgae species we find in Hawaii. And I'm going to be doing this through the application of my research methodology as a kanaka ʻŌiwi in Ainu Moser. So first, I'll give a little bit more background about all of this, but I want to first ground ourselves in Oshodo Bay. Yeah, okay, I'll just tell you next slide. Okay, so next time. Yeah. So here, this is Oshodo Bay. Hokkaido University has a marine station there since 1908. It's about an hour away from our campus and lab in Sapporo. So it's a very convenient location, but most importantly for this study, I want to look at different sources of variability, especially seasonal variability in dissolved organic carbon dynamics. And so it has very stable tidal levels. It's accessible year round, even in the winter. And very, very biodiverse ecological habitat in terms of macroalgae and different kelp species. So next slide. So here you can see the location of Oshodo Bay relative to Sapporo within the, yeah, anyways, you can see the general macroalgal ecosystem within the bay and the two main areas that Hosome kombu dominates. It dominates about a third of the total area. So that's why I'm mainly focusing on this kelp. As you can see, it grows quite densely, but there's a whole bunch of other species that grow there too. Okay, next slide. To give some environmental context, we're way up north, so we get a lot of snow in the winter, and that's why salinity is normally around 33 year-round, but it does dip in the spring due to snowmelt runoff. And in terms of the temperatures, they get as low as 5 degrees in the winter, reaching maximums of 20 to 25 degrees in the summer. One thing to note is this period of change in the spring is really critical because this change in salinity affects the osmotic cellular valve of the kelp, and that stresses the tissue and is potentially linked to the mechanisms of release for DOC. Okay, next slide. This is an annual kelp, so it's growing from a minimum biomass in the winter, and in January, increasing biomass to peak in the summer, then decreasing by fall due to erosion and grazing natural decay processes. So you can see how that peaks in the summer, but we do have quite a bit of variability. In the spring, there may be some larger kelps, but then still some smaller ones. And so it's important to think about, even within a single sampling event, what is the correlation between different biomass sizes and dissolved organic carbon. Okay, so now that we have that context, I want to go over the application of my methodology, make sure that we're all on the same page. That methodology is critically different from methods. It has to do with the theoretical lens and how we design and conduct our science. So we all have research methodologies. We all have research standpoints that are informed by the ways that we think, the ways that we are, the way our different value systems or social position. Whether or not this is explicitly stated or not, we all have our own methodologies. And then in the next slide, I argue in my methodology paper that it's critical for us as geoscientists, as conservation, environmental scientists, to try to orient our methodologies and our science towards decolonization because of not only the moral obligation to decolonize and to strive for Black and Indigenous sovereign liberation, but we have professional obligations because of the intimate relationship between colonialism and the climate crisis, the planetary crises, that if we're trying to conserve, trying to solve the climate crisis, that this is more or less futile if we're not addressing the root of this, which is colonialism. Okay, next slide. So I'm stating that if we articulate and reorient our research methodologies, that this is one way, based on our specific standpoint, positional obligations to local peoples and places, that this can have critical and material outcomes to not only how we design the research, but also how we interpret and discuss that research. And I think a lot of people are doing really great work here in Hawaii and are kind of already implicitly incorporating that without kind of going to the extent that I am to being specific about it. But I thought that it was important to be very explicit for my own PhD dissertation. Right, next slide. So as you can see here, this is how I various how applying ʻŌiwi worldviews of an which is a concept taken from Professor Jamaica Hilimele Kalani Osorio's Remembering Our Intimacies book. And so all of this informs my kulana and my kuleana as I do my research. Next slide. And sort of the outcomes of that is a clear understanding of my position as a Kanaka ʻŌiwi in Ainu Moser. So what are my obligations with that positionality? How can my research on seaweed biogeochemistry, the implications for blue carbon sequestration, potentially blue carbon credits, how do I make sure that that is advancing indigenous sovereignty and not, for example, just a perpetuation of settler colonial industry and massive scale industrial seaweed farms. Okay, next slide. So just a very brief overview of what kind of the macroalgal organic carbon cycle looks like. The macroalgae are taking up inorganic CO2 through photosynthesis, increasing their biomass, and then laterally exporting VOC in particulate and dissolved forms. A portion of this is respired back up into the atmosphere through microbial activity. That's the more labile or easily degraded forms of carbon. But the more refractory portions can persist for a long time, hundreds to thousands of years, for example, and be vertically exported below least depth, potentially being sequestered. The numbers shown here are taken from a 2016 paper, which estimated those sequestration potentials to be greater than seagrasses and mangroves and other traditional blue carbon ecosystems. But you can see quite a lot of variability in those estimates. That's because there's very limited experimental data, but on top of that, there's a lot of variability within individual studies in terms of differences in individual kelp or macroalgal samples. So in the slides that I'm going to be sharing here, I'm going to be talking about the differing roles of passive versus active exudation mechanisms of VOC release, specifically in terms of how they're related to biomass, so primary productivity, that has to do with the active exudation, again, irradiance through photosynthesis, and then seasonality, and what this can tell us, and maybe explaining some of those sources of variability. Okay, so next slide. So here I'm showing that there was a very weak relationship, kind of contrary to what we'd expect, contrary to a lot of the assumptions in the literature, that they just assume we can estimate VOC release from the amount of biomass, amount of primary production, that it's a proportional metric. This only, in my data, we're seeing biomass is only explaining about 14% of the variability in VOC release. You can have some very high biomass samples that have very low VOC release, and conversely. So this is an indication that maybe passive exudation mechanisms are more important, and there's something else going on here. Next slide. In terms of the irradiance, photosynthetically active radiation across 3 different treatments, there was no significant relationship with higher irradiance and higher DOC release. Again, another indication that passive exudation is more important. Next slide. Just showing the across the different sampling events, you can see some of the general trends of higher release during the spring and the fall. Again, especially in late March, very high individual variability. Last graph slide. Simplifying this into 4 different categories, we clearly see seasonal variability, with actually the fall and the peak decay period being kind of the most important. Statistically significantly higher than early decay and early growth, but also the mean value higher even than the peak growth period. So to recap, in the last data conclusions slide, we see the irradiance not really being a factor, the importance of passive exudation, which may be overlooked. And this is all kind of an indication that extrapolating estimates and seeking that universal generalizable answer is not really the most appropriate route for understanding the mechanism of DOC release. And last final slide, in terms of the implications for indigenous cultivation, one, on a practical basis, that harvesting kelp and maybe other fleshy macroalgal species before the natural decay period means that you're removing that very significant source of dissolved organic carbon and reducing local DOC contributions, maybe even global DOC contributions, and this coupling between the biological condition, for example, bleaching of the tissue, other sources of stress, that either DOC could be a proxy for biological and ecological health, or on the flip side, that a very healthy ecosystem could be an indication of DOC. Maybe you can estimate blue carbon based on the biological condition of the seaweed or the ecological health of the coastal area. And all these different sources of complexity and variability that I'm highlighting here are just another reminder of the importance of restoring these complex relationships, which is best achieved by restoration of indigenous governance and stewardship. Hello, and aloha. And yeah, so questions. Sorry, I know that was a lot of information. Try to pack it all in.
The INU already following this sort of procedure in their cultivation facility, or
so the situation in Hokkaido, in Japan, with indigenous Ainu people is very, very complicated. We're basically in the situation that the Japanese government has not even acknowledged them as indigenous people until sort of 2019, and kind of the histories of violence against Ainu people has made it so that many Ainu people do not feel comfortable publicly identifying themselves as Ainu. So there are Ainu communities still in Hokkaido, but we do not have the same level of like nonprofit biocultural restoration or cultivation programs as we have in Hokkaido. So there are no Ainu seaweed cultivation organizations or anything to that extent. So the discussions that I have, or the relationships that I'm building with Ainu people, is just like a person-to-person relationship. And that's one of the reasons why, as part of my methodology, I think it's so important to have this orientation, but also with the understanding that I need to build and maintain these relationships beyond my PhD program, because there's really only so much that I can do at this point.
history of them?
Historically speaking, yes, kelp especially has importance to Ainu people as food source, as a cultural source. I in my research and in my methodology, based on different discussions, I'm choosing not to try to expound on any of that information, even that that, that I've received or that I've read or heard about, because of my positionality, I don't feel comfortable kind of asserting myself as an expert on these topics, because there's a lot of problems with where some of that information comes from, where, it's like documents from the early 1900s done by Japanese anthropologists that, have a very fluent mentality, and it's, my ability to discern what is appropriate and useful, and to Ainu people, that's not my place. That's why.
Well, I think that's all we have time for, but I just want to say, like, so often the story of research here in Hawaii is that we have Western methodologies being, brought here from afar and being done. It's extremely refreshing to see an ʻŌiwi methodology not just being done in Hawaii, but actually examining relationships in the land in other places around the world. And I think that that's the first study of that kind that I've seen. So this is really exciting. Thank you so much.
Yeah, thank you.
Yeah.